Okay, hi. Sorry for the little bit of a delay, but today we are going to be talking about macromolecules. So last time we talked about chemistry of life, so chemic, basic elements that compose life, um, and what's important to know about atoms and molecules and how they interact with each other. So today we are going to be talking about how these elements and molecules come together to make larger units that are very, very important in creating and maintaining life. So let's talk about that. So what we're going to be working on are basically small molecules. They're small molecules for in cells that are important for its existence and for its function. There are four major families of small organic molecules. We talked last time about how carbon is a basic important unit of life and how all life has these carbon compounds. Um, so the four major families of organic compounds that we're going to be talking about are sugars, fatty acids, amino acids, and nucleotides. So let's get started. So these small organic building blocks that you have inside your cells and all living things essentially, they are going to come together to create larger organic molecules in the cell that can be part of their structure and function. So sugars, different types of sugars will come together to create polysaccharides, glycogen, and starch. So energy molecules and storage molecules. Fatty acids will come together to prepare membrane lipids as well as fats. Um, Amino acids will come together to make proteins, and nucleotides will come together to make nucleic acids, RNA as well as DNA. So this table shows you the basic composition of a simple cell, a bacterial cell, but that can also be taken extrapolated into mammalian cells or larger eukaryotic cells as well. Majority of the cell's weight comes from actually water, right? Because it is such a major component, because it has such important solvent properties as well as other properties inside metabolic processes that makes up 70% of the total weight of the cell. And then inorganic molecules make about 1%. And the remaining is with organic molecules, whether it be sugars, amino acids, proteins, or fats. And that's what we're going to be talking about. So the macromolecules, these big organic molecules that we just talked about that are made of these small subunits, are 24% of your mass. So the first thing we're gonna go through them one by one. The first one we're going to talk about are sugars. So sugars are your basic energy source. Their basic subunits are called monosaccharides, and when they are coming together to form long chains or structures, those are going to be polysaccharides, lots of sugar molecules stuck together. Monosaccharides are simple sugars. They just contain a simple subunit, a single unit of whatever sugar molecule we are working with. They have a very particular formula. All sugars that you're going to be looking at, they will all have the same basic formula, and then you can have it multiplied by 3, 4, 5, or 6, depending on what type of sugar you've got. The general formula that it has is a carbon and a water molecule combined together, and then you can have them as many times as you need to create the type of sugar that you're looking at. Glucose, which is the basic sugar that is utilized by our cells as the energy source, is C6H12O6. So basically, it's 6 of these general basic units combined together. Monosaccharides come in many flavors. They can be aldose or ketose. And then within each category, they can be 3 carbon, 4 carbon, 5 carbon, 6 carbon, all kinds of different things. A 3 carbon sugar is a triose. Glyceraldehyde is an example of that. 5 carbon sugar are pentose with 5, and ribose are examples of those. And then 6 carbon sugars are hexose. Glucose is a 6 carbon sugar, so it is a hexose. Fructose, another monosaccharide with 6 carbons, is another example. These sugars can combine together under certain conditions to form a ring structure. This is usually in aqueous solutions. These aldehydes or ketone groups in the sugar molecules will react to the hydroxyl group in the same chain to create a ring structure, which is what you see right here in the case of glucose and ribose. This is your first carbon. If you look, right, the carbons are numbered. So the first carbon is what is reacting with the hydroxyl end. So this is your glucose, this is your ribose molecule. So the 6th carbon is actually freestanding on the side. So the isomers basically are when your monosaccharides are appearing again in the same kind of ring structure, but the ring is formed at a different spot, kind of, right? So the hydroxyl attachment is at a different spot, and those are your isomers. Just this simple rearrangement can create a completely different sugar molecule with different functions. So these differences, these small differences, can make minor changes in the chemical properties of the sugars, but it makes a major difference in how these are recognized by our cells, by the proteins inside them, and whether or not they can be used in a metabolic process. So here, the 6 carbon sugar, the C6H12O6, depending on how the arrangement of the groups happen, you can end up with a glucose, galactose, or a mannose molecule. So in glucose, this connection is at carbon 1. In galactose, it is on carbon 4, and in mannose, it's on carbon 2. So the hydroxyl group on the carbon that carries the aldehyde or ketone, it can change. These isomers basically form, right? So it's basically them switching back and forth along that axis. This forms because it can rapidly go from one position to another, and these two positions are called alpha versus beta hydroxyl. So alpha hydroxyl right here and beta hydroxyl right here. So as soon as these sugars are linked with other, right, this particular ring structure is linked with another one of its own or another, this is frozen and it is maintained. However, when they're just by themselves, single hanging around, they can be in these various positions, and it can make them a little bit more mobile. So the sugar derivatives that form because of this hydroxyl group changing or getting replaced by other groups. So a glucose, again, molecule can have other groups attached to it to create a glucosamine when there's an amine group added to it, right? An acetylglucosamine when there's an acetyl group added to it, or glucuronic acid when there's a carboxyl there. So what we can do with this is, obviously the sugars on their own as a monosaccharide can have certain functions and can be involved in certain metabolic processes, but they can also be linked together. Many times the sugars that we see inside our cells are actually in the shape of polysaccharides. These polysaccharides form through glycosidic bonds. And here you have an example of 2 monosaccharides that are coming together through glycosidic bond to create a polysaccharide or disaccharide in this case, because there'll be 2 molecules stuck together. And then hydrolysis would be the breaking up of those polysaccharides or disaccharides into individual monomers again. So here you see an example which shows this condensation reaction happening, which will release the water molecule as these 2 groups combine together, 2 monosaccharides combine together to create a bond, and this is your disaccharide that is resulted. And then hydrolysis, where water is used to return that bond back into its original form, so you can get the two monosaccharides back out again as well. So the hydrolysis breaks up that bond, this bond right here. This is the reactive bond in this case. Disaccharides are the next category that we're going to be looking at, because again, they are extremely important in metabolic functions. So an example of that is your alpha glucose and a beta fructose coming together to create sucrose or table sugar, right? The general sugar that we eat. And this forms again using the glycosidic bond. Now, there are three common disaccharides that are important in biological functions. And those are glucose and glucose joined together, glucose disaccharide, which is maltose, the lactose and glucose coming together, creating lactose, which is the main component of milk, and then glucose and fructose coming together to create sucrose, which is your sugar. They have many functions. So now you can have a disaccharide. You can also have longer chains of sugars, multiple monomers coming together to create a longer chain. They have two main functions in the cell. One is storage and the other is structure. They are held in reserve for energy production. Glucose, the monomer, is used for actual energy production. Polysaccharides like disaccharide and beyond are held in reserve for energy production in the form of glycogen in animals and starch in plants. Structure is also provided through these polysaccharides because they create a kind of a lattice or a kind of a scaffolding or structural support. They provide mechanical support for cells. They are in the form of cellulose for plant cell wall to create that rigid structure around the membrane to give it its strength, and then chitin in insects, which is that exoskeleton. Similarly in fungal cell walls as well. Small pieces, small oligosaccharides that are different monomers that are coming together to create these polymers can be linked to other macromolecules as well, whether it be a protein or a lipid, fat or protein. And in doing so, they can provide those lipids and proteins additional functions. These also help protect the cells from recognition or from different sources, and allow them to interact and communicate with each other as well. So proteins, when they have a sugar molecule attached to it, are called glycoproteins. These are covalent linkage of sugar molecules to protein molecules. And similarly, sugars that are attached to lipids, those lipids are called glycolipids. So here is an example of some of these structures. So oligosaccharides and polysaccharides, these are again large linear structures or branch structures. They don't have to be linear or branch structures that are made from several monomers of simple sugars, monomers coming together to create longer chains, longer structures. Short chains of these are called oligosaccharides or polysaccharides. Glycogen is a common example of such polysaccharides that's entirely made of glucose molecules, and it's a great storage molecule for cells. So here is an example of glycogen. These are all glucose molecules that are stuck together in different ways to create branch structure or to create long linear chains. And that's an example of a structural support molecule in there, not structural support, but rather a storage molecule, but provides some structure as well. Next up, we're going to be talking about fatty acids. Fatty acids are going to be amphipathic, which means that they are not going to be like, some of the other molecules that we are going to look at that have only a hydrophilic or hydrophobic region. You know how many times we talk about how a molecule is polar or non-polar only. Well, fatty acid has both a hydrophilic or water-loving area and a hydrophobic region as well. It has a hydrophilic carboxylic acid head, and then it has a long carbon, some type of hydrocarbon chain, whatever length it is, that's going to be hydrophobic. And that's just a simple fatty acid. There are several different types of fatty acids that are important in our cells. One of them is triacylglycerols, which are your storage lipids. In this case, you have three fatty acid chains that are combined together with, or they are joined together with a glycerol head. The glycerol molecule is going to be your hydrophilic head, that's your polar side, and then your hydrocarbon chains are going to be your hydrophobic side. But these triacylglycerols are the main energy reserve inside the cells. These are the fatty acids, and they are joined with your hydrocarbon chains with glycerol using an ester linkage to form a triacylglycerol. These are going to be different structurally depending upon the length as well as the saturation of the fatty acid that is being attached to that glycerol. In a saturated fatty acid, the hydrocarbon chain has no double bonds. It's completely saturated with hydrogens so that there is no double carbon-carbon bond. And that makes for a more compact, more sleek looking hydroglycerol, or in this case, a triacylglycerol. However, if you have an unsaturated fatty acid, that means that it is not fully saturated with hydrogen atoms. And so there is some place in there where there is a carbon-carbon double bond that will lead to a kink in the chain, in the hydrocarbon chain. And those kinks are going to give it a little bit more bulky appearance and bulky structure, and wider structure. So phospholipids that make up our membranes, all our bilayers, lipid bilayers of our cells, are important in this membrane structure, and they also occur very similar to this triacylglycerol structure that we talked about. They again have hydrocarbon tails, but this time there are only two of them, and this time they're joined together in the hydrophilic head with a phosphate. So they are phospholipids because they have that phosphate group that is attached to these hydrophobic fatty acid tails. This hydrophilic head, this phosphate, can further be changed by adding a choline group to it as well, in which case it is a phosphatidyl choline. But in a typical membrane, the main component is going to be the phospholipids that are there. You can also have other polar groups attached to it as well. It doesn't have to be a choline only. And they can have sugars attached to it as well, which would make you a glycolipid. So here is your basic structure of a lipid bilayer. You have your fatty acid chains, your glycerol, your phosphates, and then some polar group in many cases that is attached to that phosphate group. The difference here is that it's a 2 hydrophobic fatty acid tails instead of 3 that are joined together to that hydrophilic head. Now again, these membranes, because there's an aqueous environment both inside as well as outside the cell, are going to require some buffer zone, and that is found in our membranes because our membranes are lipid bilayer, where these phosphate groups interact with the aqueous environment outside and inside the cell, and the lipids tails face each other to create that hydrophobic region, to create that nice clean barrier. The phospholipids, like I said, can have many things attached to them, and depending on what's getting attached to them, they'll have different names, right? And so you can have phosphoglycerides, or you can have an amine group attached to it. You can have sphingomyelin, so there are different molecules that can be attached to it. All of these are called phospholipids, whether they have a glyceride or an inositol or an amine, whatever group they have, but they will have different names depending on what type of group is getting attached to them, or if they have sphingosine, which is the sphingomyelin. And then you have your glycolipids, which have sugar molecules attached to it, and you can have different types of sugars attached to it. And they can be attached to it using O linkage or N, so they could be O or N linked glycosylations. Now, lipids, anytime you have lipids and you put them in some type of an aqueous environment, are going to form some aggregates to create a stable structure. So they could create a surface film where the phospholipid heads are interacting with the water and the tails, the hydrophobic tails, are sticking out. They can create micelles where they kind of just, all the lipid tails, the hydrophobic tails, are interacting inward, keeping the water molecules out and creating these ring structures or round ball structures. They can also create other things as well. They can form larger aggregates also. So in a phospholipid and glycolipid, they form self-sealing lipid bilayers, and these are what we called our lipid membranes. These are your membrane that make up the outer part of the cell, as well as the nuclear membrane and a lot of other structures inside. These lipid aggregates, like I said, will form anytime spontaneously as well if they're put in an aqueous structure. Triacyl glycerols, for example, will form large spherical fat droplets in the cell or the cytoplasm, and you can actually identify them inside it when they're inside the cell. Now, lipids, just like sugars, can also form ring structures under certain conditions. Steroids are an example of such types of lipids. They have a common multiple ring structure. So this basic structure right here, this is the structure of a steroid. This structure can then have other reactive groups attached to it to make it different types of molecules. These molecules are extremely important in our cells to help create different hormones, to have different functions. Cholesterol is one of these molecules that is found in many cell membranes, and it prevents our membranes, it keeps our membranes more fluid and prevents them from becoming too rigid. You can also have testosterone, which is the male sex hormone, estrogen. Many types of hormones are actually off of the steroid structure as their basic part. Now, one thing that is I would like to mention in there is, again, your lipid, these tails, the hydrophobic tails, again, can be fully saturated or can be unsaturated or be a mixture of both. A cell membrane from a particular cell or a particular animal or a particular structure doesn't all have to be at the same composition of saturated or unsaturated lipids. These the abundance of a particular type, saturated versus unsaturated, changes with the environment that the cell is in or that the organism is in, the diet that they are taking, their needs for adapting to a certain conditions. All those play a role in how much of those hydrophobic layers are going to be saturated versus unsaturated. So when you have a lipid membrane that is composed of more saturated fats, it's going to be more rigid, right? And when it is going to have double bonds, it's going to have these kinks, so it's going to be more fluid and be able to be more flexible in that environment. So that is something important to be aware of. The next thing we're going to talk about are amino acids that make up our proteins. Amino acids are the basic subunits that make up proteins. They again have a general basic formula. So you will notice for every single macromolecule, so for a sugar, you had monosaccharide as your basic unit, and you had the polysaccharides as your macromolecules. For your fats, you had the fatty acids as your basic unit, and then you had the hydrocarbon chains, the glycerols, and your phospholipids as your molecules. And here you have amino acids as your basic structure. The lipids mostly were made of that carbon hydrogen, right? Here you have another group, you have that amine group. So, and you have an R group. So the general formula for an amino acid is this one, these carbon that is attached to a carboxyl on one end, a hydrogen on the other, an amine group on the third, and then there is a side chain, which is your R group. The R group is common, is going to be different for your different amino acids. There are 20 different amino acids commonly, and those are your 20 different side chains. At pH 7, both the amino as well as the carboxy end of the structure is going to be ionized, so they can both be reactive. So parts of an amino acid, again, we talked about the amino group, the carboxyl group. This is your alpha carbon, and then you have the R group and the hydrogen. In a non-ionized state, your R group in this case, right, it has the CH3 on it. If you put it under pH 7, it is going to become ionized, so you'll have the carboxyl and the amino group as ionized. So that helps it then become more reactive and interact with other groups. The alpha carbon is symmetric, and it allows for two mirror image isomers, L and D isomers for each amino acid. So you have these L versus the D amino acids. Proteins that are inside our cells contain exclusively this L amino acid. Those are the isoforms that are used. Amino acids are in a protein also formed through condensation reaction. They're held together by what we call a peptide bond. That's just the name that we utilize, but it works pretty much the same way. The N terminus of your polypeptide over here and the C terminus of your polypeptide over here, where it is composed of multiple amino acids joined together through peptide bonds, which are formed through, again, a condensation reaction. Right here, you can see this example of that peptide bond. And here you have your different R groups that make up the different amino acids. This one is phenylalanine, serine, glutamine, and lysine. There are 4 basic families of amino acids. That's based on what side chains are there and what kind of properties they can have. They can be acidic, basic, uncharged, polar, or non-polar. And depending upon these properties, the final protein is going to have certain properties of interacting with other molecules or being certain things. Each of these 20 amino acids have been given a 3-letter acronym as well as a 1-letter abbreviation. For example, alanine can also be written as ALA or just simply as a capital letter A. So for each one of these categories, I'm just going to show you some amino acids that are the important amino acids that are part of it. So amino acids with the basic side chains, and we expect you to know these, are lysine, arginine, and histidine. These are all going to be basic under a neutral pH. And this group is extremely basic because it has a positive charge over here that is going to be stabilized in this case by resonance, and in this case because the nitrogens have a relatively weak affinity for the hydrogen protons, and they are only going to be partially positive at neutral pH. Another one is going to be now, obviously because we have these basic, you also have some very acidic side chains, and those are your aspartic acid and the glutamic acid. So this is ASPD or glutamine, which can also be written as E. And then for your uncharged polar side chains, we have asparagine and glutamine. These are not, these are polar even though they are not charged. And then you can, usually when you have either an amine group or a hydroxyl group, those are all uncharged but polar, or they may not be uncharged, but they are polar side chains. So in this case, even though the amide is not charged, it's still going to be polar. And in this case, even though the hydroxyl is not ionized or charged, it's still going to be polar. So asparagine and glutamine are both uncharged polar side chains because of this amide N that is present here, and serine, threonine, and tyrosine have the hydroxyl in their side chains that makes them polar as well. And then the bulk of the remaining are non-polar side chains, and these are your alanine, valine, methionine, tryptophan. All of these, if you look at them, are either, composed of just carbon and hydrogen, or even when they have a ring structure or another compound, it's still pretty non-polar. So here you have those: proline, phenylalanine, leucine, isoleucine. Those are all mostly carbon-hydrogen chains. And then here the glycine, cysteine, methionine, and tryptophan, while they have other things like sulfur and nitrogen attached in there, they are no longer polar because of the other properties there. There is some of these, for example, cysteine can form disulfide bonds between two cysteine side chains next to each other. So they can have some reaction because of that with this disulfide, and they can create disulfide bridges because of that. Finally, the last macromolecule category that we have are nucleotides. Nucleotides are the subunits of DNA and RNA. So at the end of the day, if you look at these molecules, right, three of them, they're all storage molecules in some way, shape, or form. Fatty acids, your proteins, as well as your sugars, they are all storage molecules for energy. Nucleotides, on the other hand, you'll be like, well, that doesn't store any energy as such. Well, it does in the form of ATP, but also it is a storage molecule for information. Nucleotides are the basic subunits of DNA and RNA. They are made up of bases. These nucleotide bases, there are 5 that you would need to know. There are 3 pyrimidine, uracil, cytosine, and thymine, and 2 purines, which are the 2 ring structures that are the adenine and guanine. Now, depending on whether you have a uracil or thymine, it is going to be a DNA or RNA. Uracil is only found in RNA, and thymine is only found in DNA. The nucleotides, again, have a basic structure. This time it's getting a little bit more complex. It has the basic sugar. This time it's a pentose, a 5-carbon sugar that is attached to a phosphate group that gives it a negative charge. And then it has on carbon 1, it has a base, and that base is either your pyrimidine or purine. This is your basic nucleotide, and these are the basic units of nucleic acid. These nucleotides can be short-term carriers of energy, of course, chemical energy in the form of ATP and ADP, which is, as you know, the basic currency of energy inside our cells. There's adenosine triphosphate, which has these 3 phosphate groups linked together. That makes it a really highly reactive, high-energy bond that can be broken up to release energy that can then be utilized for metabolic processes. So adenine with a ribose and then attached to the 3 phosphates. This energy is then utilized for cellular work and for chemical synthesis of stuff. These nucleotides are joined together via phosphodiester bonds in DNA and RNA, and that's right here. You can see that it's a phosphate group that's attaching to the 5th carbon or 4th carbon, and that is what is going to give you that phosphodiester bond. And at the 5' end, you'll have a phosphate. At the 3' end, you'll have this oxygen that's open. And that's what's going to give you the long chain of DNA or RNA. In this case, you have a guanine, adenine, thymine, and cytosine joined together. The nomenclature for these is important. The basic base is just known by the simple name of the base itself: adenine, guanine, cytosine, uracil, and thymine. When they are attached to the sugar, they become a nucleoside. So base plus sugar is a nucleoside, and they will be adenosine, guanosine, cytidine, uridine, and thymidine. And they're abbreviated by AGCUNT. And when that base and sugar is also attached to a phosphate group, they become a nucleotide. And that would be, again, your next thing. So adenosine triphosphate is going to be obviously attached to that phosphate group as well. Uridine diphosphate, because it has that phosphate attached to it. Adenosine monophosphate, that tells you how many phosphate groups it's attached to. Mono for 1, di for 2, tri for 3. So these nucleotides have other functions as well. They don't just make DNA and RNA. The di and triphosphates carry chemical energy. Obviously, they can also be used for energy storage. They can combine with other molecules to form enzymes, like coenzyme A. That's an example of that. They can also be used in chemical signaling processes, intracellular signaling processes. Cyclic AMP is an example of that. So just to review, macromolecules in our cells, each macromolecule contains a very specific basic structure that is then utilized to create sequence of subunits. Non-covalent bonds are used to specify the precise shape of a particular macromolecule, and non-covalent bonds also allow the macromolecules to bind with other selected molecules. Going back to our cells, remember that 70% of our cells is just water, and then that remaining 30% is going to contain, majority of that is going to come from those macromolecules, about 24%, where it is the DNA, RNA, protein, and polysaccharides, and then the remaining small amount, remaining 4% or so, is going to be from the inorganic ions and small molecules. The subunits for sugar, again, is your monosaccharide, and macromolecule is called a polysaccharide. Amino acid is your basic subunit for your proteins. Nucleotide is the basic unit for nucleic acid, which is your DNA and RNA. As you grow these macromolecules, you do that by doing condensation reaction, where the subunit hydroxyl end is joined with the hydrogen end of the growing polymer to release a water molecule and make an elongated chain. And then it can be, water is used in hydrolysis, through hydrolysis to break up those bonds to release those monomers back out. These molecules can then, these macromolecules, obviously are formed through this very precise reaction. However, when they are in different environments, they can change their shape and structure based on non-covalent bondings. These are partly due to these individual molecules in their structure, right? So the R groups in a protein can dictate how they are going to be folded together in different pH or different environments, under hot versus cold, under pH 7 versus pH 5. In each environment, they'll have a stable folded conformation, which can get disrupted under other conditions to create an unstructured polymer chain. Obviously, our goals are usually in the cell to maintain a particular structure so they can perform a particular function. These non-covalent chemical bonds that are forming are because of the weak non-covalent forces. A lot of it is from those hydrogen bonding and van der Waals forces that we talked about last time. So remember hydrogen bondings, and that's actually how DNA is maintained in the double helix, is through those hydrogen bond interactions between the purines and the pyrimidines that are joining these molecules together and keeping that those two strands of DNA together, as we'll learn about in the later chapters. Hydrogen bonding also allows these structures in proteins to have certain structures form in the polypeptide chain, especially under the aqueous environment. Other forces that play in here are the electrostatic forces due to those partial polar bonds that are present in the R groups or in different molecules or proteins, whether it's proteins or fats or whether it is a sugar. And these electrostatic interactions will allow different enzymes to function in just the right way, making their substrate available to the active site in just the right conformation. These macromolecules can change, interact with different other molecules that they can interact with, and depending upon how strongly they can interact with them, because of these non-covalent forces, they can have bindings with stronger bindings with certain molecules compared to others. these subunits not only make single macromolecules, but these different types of macromolecules can also come together to create larger structure. An example of this is ribosome, where it is composed both of RNA as well as protein molecules. These amino acids and nucleotides individually are going to make the RNA and proteins that are part of that final structure. And then those non-covalent forces will join them together into that final ribosomal complex that forms. So that's all for this one. Please let me know any questions that you have about this content, and I can try to answer them for you in the comment section.